So what I'm going to try to do is to, yeah, that's chat. Okay, I'm going to hide the chat. I'm going to hide the participants. Okay, I'm going to try and record a kind of a video full chart of the class. I hope it will make it more structured. So I usually suggest to learn them starting with the main mechanisms. So we've got here, let me make it the font size bigger. Okay, probably. Old cell wall inhibitors. Okay. Then we have ribosome inhibitors, protein synthesis inhibitors. We're going to put it somewhere here, and then we have acid inhibitors, and then we have double. So when you structure it like this, I think it makes more sense, sort of. So cell wall. What about cell wall inhibitors? So what we've got here, we have three big groups. We have beta-lactams, right? We have glycopeptides. And we have separate kind of a class, basitracin. Okay. So in beta-lactams, okay, let me change it a little bit. I'm going to draw the lines. So beta-lactams, glycopeptides. Beta-lactams, we're going to have the following. We're going to have, so what's the mechanism for supposed to good. So what do we have for beta-lactams as the mechanism? Beta-lactams, we have inhibition, the cell wall of the peptide linkage synthesis. Placing down here. Move. Okay, so what are the classes of beta-lactams that we may remember? Okay, Marlon, how you doing? We have penicillins, sporins, monobactams, carbapenems. Okay. So glycopeptides, tracing. Okay, we're good. Now, what is the mechanism for glycopeptides? It inhibits the inhibition of peptide linkage synthesis and of synthesis. Good. Basitracin, it's going to be inhibition of the transport of the wall components. Okay, so that covers the cell wall inhibitors. I'm going to try and make it look nicer. Line, here's line. That's your cell wall inhibitors. Now, protein synthesis inhibitors. We have quite a few here. I don't know if we will be able to fit it all on the screen. If not, we'll just make like a separate thing for them. Protein synthesis inhibitors, we have two big types. One is 30S subunit inhibitors. By the way, if you have questions, do not hesitate to like interrupt me, okay? And under this one, we've got two more. We've got two classes. One class is aminoglycosides, and another class is cyclins. Aminoglycosides, the mechanism will be what's the word? When it distortion of the ribosomal structure of faulty. Okay. For tetracyclines, inhibition of tRNA binding. Okay. So far, does that make sense, folks? Tell me. Okay. Well, I guess that makes sense then. I mean, really, because I cannot see your faces, your cameras are off. I'm gonna kill this too for now. Okay, and then we have 30S, 50S, sorry. 50S inhibitors, which can be further divided into, there's one group that includes glucosamides, streptogramins, and macrolides. Another class of these antibiotics is cephalosporins. I'm going to move this thing here. And another class will be sazolidinones. So lincosamides and macrolides, they prevent block release of peptide chain from the 50S subunit. Cephalosporins inhibit the formation of the peptide bond between the incoming acid and the growing 10-hexazolidinone inhibit the formation of the initiation complex. Okay? So we try and, okay, good. So we're going to make it nice. The arrows. So we've got here protein 30S subunit, 50S subunit. 30S subunit are These ones, 50th subunits are these ones. Okay. Ladies and ladies. Does that make sense?
Yes.
Okay. And remember, it all is being recorded now. So I'm going to post it today on the YouTube channel.
Okay.
Good. Now, since I'm out of space, I'm making a new document. Okay. How am I supposed to do it? Yes. Nah, I don't really need to save. Yeah, good. So recorded. Okay, now we have two more big types. One big type is, oh, I forgot one class. We'll talk about it. So it's inhibitors. And okay, let's start with nucleic acid inhibitors. So we've got fluoroquinolones, rifamycins, and nitroimidazoles. So fluoroquinolones inhibit DNA gyrase and DNA replication. Rifamycins inhibit RNA polymerase and transcription. Nitroimidazoles, for what we know, they lead to, let's put it this way, may lead to DNA degradation, may inhibit DNA. Okay, that makes it for nucleic acid inhibitors. Here, and here. Here's, I'm going to do one thing. I'm sorry. What if? Nope. Nope. Color change doesn't work. Nope. Never mind. Okay. So next one will be membrane disrupting drugs. That will be polymyxins, peptides. Well, that's kind of self-explanatory thing. They disrupt the membrane. Okay. And the last bit here will be metabolic inhibitors. So that's going to be sulfa drugs, trimethoprim, um, niacid, and arylquinolones. So sulfa drugs, trimethoprim, competitively inhibits two steps of the nucleotide synthesis. Folic acid pathway. Isoniazid inhibits glycolic acid synthesis, and diarylquinolones inhibit bacterial ATP synthase. To conclude it with a huge exclamation mark. Lines. Before we cut the recording, is that okay? Follow me throughout this thing. I think it was pretty nicely paced.
It makes sense.
It makes sense. Okay, good. So I'm going to stop recording.